I would now like to move on to introducing our next speaker, who is Mr. Arun Panchapakesan. He is currently pursuing a PhD program at the Jawaharlal Nehru Centre for Advanced Scientific Research, Bangalore, under the guidance of Professor Udaykumar Ranga, who is one of India's leading virologists. His primary field of study is HIV-1C Reverse Transcriptase, which I have no idea what is, and its role in viral evolution. So, Mr. Arun, if you could first please explain to us what is HIV-1C Reverse Transcriptase, we'd be eternally grateful to you.
Hi, am I audible?
Yes.
Yeah, so HIV is, as everybody knows, it's a virus that is responsible for AIDS. There are multiple different families of this virus, they're labeled A, B, C, D, and so on. India is infected by the C family of the virus, which causes about 95% of India's infections. And so naturally, our lab being in India studies that C family of HIV. So HIV-1C basically means the C family of HIV. And I work on reverse transcriptase, which is a protein that HIV makes. It's a protein that is unique to the family of viruses that HIV belongs to. It's what makes HIV one of the most dangerous diseases that is there. I mean, I would, if you ask me to compare as a virologist, as a person who studies viruses and makes vaccines or anything like that for them, SARS-CoV-2 is nothing compared to HIV. HIV is the ultimate challenge. If you manage to break it, you know that you've won. It's a great feeling. So it's the ultimate prize for any virologist. So that's effectively what I do here. Although I'm not trying to cure HIV, I'm actually trying to see why HIV-C is different. So yeah, I would like to share my screen. 1 second. Yeah, is my screen visible to everyone?
Yes, we all can see the screen that you shared.
Yeah. So, before I start, Professor Jayaprakash mentioned a real-time PCR instrument. That's that instrument right behind me there. I don't know if you can see it. That's the machine that has become indispensable in our detection of COVID-19. It's a very fancy instrument, and it's the reason we could detect most of the COVID-19 cases in the world. Anyway, so coming back to my presentation, before I begin, I would like to thank Ms. Shukla Bose and my friend Prem from the Parikrma Foundation for giving me this chance to talk about the biology of bacteria and viruses and how they cause pandemics. The answer to this question of how they cause pandemics is actually very simple if we understand the biology of bacteria and viruses. And I will be taking you through, I will be putting you into the shoes of those scientists because of whom we understood what these organisms are and what they mean to us, and why is it that they cause disease in humans. But before I do that, I know that Professor Jayaprakash has already taken you through the pandemics. I've given you a brief history of all the pandemics, but I would like to just list out the worst pandemics. If you see what has caused, the maximum amount of damage to human life, SARS-CoV-2 is not in the top 3. It's not even in the top 5 among history's worst pandemics. If you want me to list them, you'll find, so I have here the number, the name of the disease, what years did it affect us, and how many people did it kill. At the 10th place, we have something called the Hong Kong flu that affected in between 68 and 69, killed about 10 lakh people. Then we had the Russian flu, which infected around the late 1800s, killed again about 10 lakh people. We have the cholera pandemic that lasted quite a long time from 1899 till 1923, that killed around 15 lakh people. We have the Asian flu, 57-58, killing 20 lakh people. This is where we are right now, the current SARS-CoV-2 pandemic that has killed about 26 lakh people. This figure is as of yesterday. This is still ongoing, but it's unlikely to go to 5th spot because at 5th we have the modern plague, which has killed about 1 crore people in 1894 to 1902. So these were happening parallelly, the 6th cholera and the modern plague. Then we have the famous, everybody would have heard about this when the current pandemic started, the 1918 Spanish flu. It killed about 2 crore people. That was about infected 1/3 of the world's population at that time and killed about 2 crore people. Then we have what I'm working on, HIV/AIDS from 1960 till today. It has killed about 3.9 crore people. We have the Black Plague, which was from, the famous Black Plague, which infected Europe in 1350s. That killed 5 crore people. It wiped out 1/3 of the population of Europe at that time. And finally, the worst pandemic that we have ever had was the plague of Justinian in the years 541 AD. That killed about 10 crore people around the world. Now, if we look at this table, you'll find that about 4 of these 10 diseases are caused by bacteria, and the remaining are all viral diseases. There's another thing that this table is telling us, which is that there's something that happened that after 1923, which is the 6th cholera pandemic, there has not been a major bacterial pandemic. There have been small outbreaks, but we've always managed to contain them because before they became something huge. And that is because of something that happened in 1928. Something magical happened, and that has changed the course of medical history. I will come to 1928, but before that, we need to go and understand about what bacteria are and why is it that we must study them. So let me ask you a question. Are you human? The reason I ask you this question is, if you ask as a scientist what makes you human, I would say, you have human cells in your body, you have human DNA. So in or on your body, there are approximately 30 trillion cells. That's 3 followed by 13 zeros. 30 trillion human cells make up your body and make up who you are. That's an interesting point because in or on your body, there are also 30 trillion bacterial cells present at any given point of time. Right now, as I speak, I'm enveloped in bacteria. They're inside our gut, they're everywhere. So if there are 30 trillion human cells and 30 trillion bacterial cells, can you really call yourselves human? The second question is that I want to ask is, if there are the same number of human cells as bacteria, we can see us as human. Why can't we see, the bacteria? And the reason is everybody knows they're too small. But what I want to emphasize here is, do we really know how small they are? To give you an example, I've taken a few everyday objects and I've magnified them to make you understand how small bacteria actually are. For example, if this is a grain of sand that you can see there, I put my little finger next to the grain, and this is magnified 4 times, right? You can see that my finger is big, so that's magnified. Obviously, this is from under a microscope. You can see the single grain of sand magnified 4 times. If I take that grain and magnify it 50 times more, it would appear about that big. The same thing I can show you with a single crystal of copper sulfate. The copper sulfate is a chemical that's blue in color. The reason I picked copper sulfate is because in a white background, it's visible very clearly. This is about the same size as a grain of salt. If I magnify that 50 times, it looks about that big, right? Or to give you guys more perspective, that's my index finger, left index finger. That's normally, I've just kept it under the camera and taken a picture. I've magnified it 4 times here, okay? And I've magnified that small portion about 50 times. Each of these individual lines that you can see are my fingerprints. So you can actually hold up your own finger and look at your fingerprints to compare how much I've actually zoomed using my microscope in that picture. And what is interesting is, even in that picture, we cannot see any bacteria. Even when I magnify something 50 times, I make something that huge that my fingerprints are now individual lines on that screen, we can still not see bacteria. And that's because you need to magnify something at least 1,000 times to see most bacteria. You can see that this image is magnified 50 times. You need to magnify something 1,000 times to see most bacteria. And this is where the story of bacteria actually begins. We, as humanity progressed across, the mathematics was one of the first branches of science. Field picked up very late, and that was because while we had a lot of people dying and getting diseases, nobody really realized that these were actually caused by extremely small bacteria, extremely small organisms that are, causing trouble. So until a Dutch cloth merchant, this is where the story of microorganisms begins. In 1676, a Dutch cloth merchant was really bored. He started making lenses, and he liked looking at things bigger, so he kept making better and better and better lenses until one day he made this. This was the world's first microscope. It's a very simple device. There's a small lens there. You would put a drop of water at the end of this pin here, and you can look at it from the other end. And when he did this gentleman, he found that that drop of water was filled with a lot of tiny moving organisms, and that was the first time anybody had ever observed bacteria. Until humanity had been around for at least 50-60,000 years as human beings, but we had never seen a bacteria until this man Antony van Leeuwenhoek came in and looked through this microscope and saw that there are organisms that are living at that small scale also. Once Leeuwenhoek had made this, the field of bacteriology took a change after about 200 years. For about 200 years, 150 years, nothing much happened. Then these three gentlemen came along and they changed again. They laid out a lot of principles. These gentlemen proved that it's the bacteria that causes the disease. Many of the diseases that we know today are actually originally identified this long back, mainly by these two gentlemen, Pasteur and Koch. Koch was responsible for identification of the cause of tuberculosis, cholera, all of the diseases. They found that many of the ailments that people were suffering from at that time, you could actually pinpoint the bacteria that was responsible for it simply by looking under a microscope. And they went much further. They had a lot of theories that they put forward, especially Koch here. He did a lot of experiments. One very simple thing that he said or did was, if a person is sick and you take, you can actually take the bacteria that causes the disease from him, and if you put it into a healthy individual, that healthy individual is going to become sick. It's a very simple rule that Koch made that changed the course of medical history. So because of them, we now have some very fancy, famous pictures of bacteria. We have these, so these pictures are all electron micrographs. These are magnified. So you need minimum 1,000. These are magnified at least 100,000 times, a lakh times through an electron microscope. So because of that, we can see this is Mycobacterium tuberculosis, the bacteria that causes TB. We have cholerae that causes Vibrio cholerae, which causes cholera. We have Staphylococcus aureus that causes all of those pimples and other boils on the faces. We have Yersinia pestis, the organism responsible for the plague. We have Lactobacillus acidophilus, the organism found in curd. Curd is mainly just Lactobacillus. It's just a big blob of bacteria that we eat. It's Lactobacillus there. And of course, the famous E. coli, Escherichia coli. This is what all of us use in molecular biology labs for our work. It's impossible to imagine a molecular biology lab without this bacteria. So what is inside a bacteria? It's very simple. They're the simplest organisms. They just have an outer wall. They have their DNA, and any food that the bacteria takes just diffuses from inside the cell. It's like if you take raisins and put it in water, the raisin swells up because the water enters the raisin, it diffuses inside. Like that, food also just simply diffuses inside through the bacterial membrane. Bacteria are very simple organisms. Some of them have this big flagellum, some of them don't. They use this for moving here and there, and their bacterial life cycle is very simple. All they do is assimilate food and then divide. That's it. They have no other objective in their life. It's just get food, and divide, and that's all they do. Okay. Then if that's the question, then it begs the question, why do bacteria infect us? And there's actually only one very simple answer for it, and that's because of food. We provide them with all the nutrition that they require to, eat food, divide that cycle, and they get it from us. Now, some bacteria, we have very healthy relationship with some of these bacteria, such as, lactobacillus, bifidobacterium. All of these bacteria actually live inside our gut, our intestine, and they help us digest the food. They help us make our vitamins. So these bacteria are all what are called symbiotic bacteria, in the sense that they are all going to help us make our food. And in return, we let them live inside our gut. They help us digest our food, and in return, we give them the food that they want. There are also other kinds of bacteria which are called parasitic, which don't give us anything. Oh, by the way, many animals like horses, they can't digest leaves by themselves. Their bodies don't have the enzymes that you need to break down the cellulose in leaves that they eat. It's only because of these gut bacteria that they can digest. Many herbivore animals use the ability of these bacteria to break down complex material like leaves to actually digest them. They can't do it by themselves. So coming to parasitic organisms, there are many of them. These organisms are in some sense selfish. They don't give us anything. They only come, take, use up our food, and in exchange, they also infect us and cause disease as a part of their process of eat and divide. I already listed a few in the previous slide. The same ones are here. Mycobacterium, you have Vibrio, Salmonella, Yersinia. All of these are parasitic organisms. There is actually a third group which falls somewhere in the middle. They're called opportunists. They will live inside you, until they see that that the time is right for them to infect. Escherichia coli falls into that group. It can cause urinary tract infections. But anyway, the focus of our talk today is actually this group of organisms, these parasitic organisms, and how is it that we manage to, they've been infecting us for a lot of years now, like easily 50, 60,000 years, these organisms have been there. They've been infecting us, but it's only in the last 100, 150 years that we actually know how to combat them. And how did we do it? It all started with this man, Paul Ehrlich. He propagated this idea of something called a magic bullet. He said, I want a bullet that can go through the host and kill only the bacteria, but not affect the host. In the sense, many medicines that we take today have a lot of side effects. Those side effects are because the medicine will, of course, work against the disease, but it will also do something to the host. That is us, right? So Paul Ehrlich he worked very hard to try to find that drug, that compound that can only go inside the body, kill the target bacteria, and not do anything to the host. His main organism was this bacteria. It's called Treponema pallidum. It causes a disease called syphilis. And Paul Ehrlich in 1909, although he couldn't exactly get his idea of a magic bullet, he did get something, and that was called arsphenamine. He made a chemical that would treat syphilis, but it had a little bit of side effects. But still, Paul Ehrlich was very famous. This was the world's first therapeutic agent. Nowadays, you use chemotherapy mainly for cancer, but back in the day, 1909, chemotherapy means using a chemical as a therapy. This was the first sort of chemical that was used to treat syphilis again, and Paul Ehrlich developed it against this. But people were still not happy because this, first of all, arsphenamine did not work against all the diseases. It was good against syphilis, but it did not work against all the diseases that were there. So the world really needed something else, and which brings us to that magical year, which I said, 1928, when Sir Alexander Fleming, many of you must know this, he was actually a scientist in the laboratory, and he got very angry, and he said, no, I'm done with my work. I'm very angry. Nothing is working. And he went on a holiday. When he came back, he saw that there was a Petri plate. So we grow bacteria in Petri plates. I'll show you one soon. And there was a Petri plate full of, bacterial colonies, and they were very old. They were from the time that he had worked on them, 1 month before his vacation. And he was about to throw them. He said, I'm waste. I don't want these Petri plates. I'm going to throw them. When he noticed something very interesting, what he noticed was this. The yellow, so this circular thing is a Petri plate. The yellow, all these are all small bacteria. So each dot there you see contains about a billion bacteria. That's why you are able to see them. You cannot see them. You cannot see one bacteria, but you can see a billion of them. And each yellow dot is a billion bacteria. He found that the plate was full of them, except for the middle where there was actually a fungus growing. What was interesting was, this is a very common occurrence in a lab. Many times when you grow bacteria, sometimes the small fungus will contaminate your cultures, and you can see something like this very frequently in the lab. But what was interesting was, even though the fungus was only growing here, there was a zone in between the bacteria and the fungus where there was no bacteria or fungus that grew. Normally, most of us would just take such a plate, not even bother to look at it and throw. But Sir Alexander Fleming asked the question, why is the bacteria not, there is space here. Why is the bacteria not occupying the space? Why is it not growing there? And Sir Alexander concluded that it was because this fungus was actually making something that killed the bacteria around it. And that this fungus was, of course, the famous Penicillium notatum, and that was the birth of the world's first antibiotic. And from then on, medical science has really changed to such an extent that now we have a big list of antibiotics. Penicillium was only the beginning. We have streptomycin, erythromycin, chloramphenicol, you name it. We have antibiotics against almost every single kind of bacteria. And that's why, more or less, bacterial pandemics are under control today. In the sense, yeah, you may have, I'm not saying you won't get a bacterial infection. I'm saying they're unlikely to cause big pandemics. And even if they do cause, you always have a treatment for them. You always know that your antibiotics would work. There's a separate problem with antibiotics, which is upcoming now because for everything we are taking antibiotics. Slowly we are getting antibiotic resistant bacteria, but that's a separate problem. By the time scientists are working on this problem of antibiotic resistance, but for the time being, bacterial diseases, we have a treatment. We know, thanks to Sir Alexander Fleming and his discovery of penicillin, we have ways of dealing with bacteria right now. So if you go back to that table, that's what happened in 1928, and which, that's why, in this table after 1923, you're not seeing any major bacterial disease. There is, of course, yes, there was also a 7th cholera pandemic, but it wasn't as bad as the other ones previously. It was mostly after 23, you will see that it's only the viruses that have caused the Hong Kong flu, Asian flu, Spanish flu, and AIDS, and of course now SARS-CoV-2. Okay, so while these three gentlemen were, I will go back to this slide. While these three gentlemen were telling the world that bacteria cause most of the diseases, we have everything, I mentioned that Robert Koch had a very simple technique. He said, if I have a diseased animal or organism, I should be able to take the bacteria that causes the disease, and if I put it into a healthy person, that person should develop the disease. Very simple rule. Almost all diseases follow this. That's what happened in the 1870s. There was a disease of the tobacco plant. This is the healthy leaf, and the leaves used to form this mosaic, and so the disease was called the tobacco mosaic disease. It was a disease that used to kill a large number of tobacco plants in Europe at that point of time. And many people were, once these three had established that you have bacteria responsible for everything, everybody went out to identify bacteria that cause. So there were people who said, okay, which bacteria causes this? And there was this gentleman, his name is Martinus Beijerinck. In 1898, he was working on the diseased leaf, and he said, okay, let me try out what Koch said. He took an infected leaf, okay, with all those marks on it, and he crushed it. Added a little bit of water, took a pestle and mortar, and nicely crushed it. After crushing it, the next thing he did was he put it into this filter that will remove all the bacteria. So if a bacteria is causing the disease, it's going to be removed from all of this. Okay. Then after removing the bacteria, he put a few drops of that purified crushed leaf extract and put it on a new leaf. Now there should be no disease, because if bacteria is causing the disease, he crushed everything and then he put it through this filter, and that filter should remove all the bacteria. But when Bajring put it on a fresh leaf, he found that the leaf after 2-3 days still got disease. Now he didn't know what it meant at that time. At that time, everybody thought it's only bacteria that caused, but he knew that it wasn't bacteria because he's removed it. So he called it something in Latin. He said there is something contagium vividum fluidum. That's what he called it. That's Latin for a contagious. There's something wrong. Yeah, a contagious live fluid. That's what it means. A contagious live fluid. Now this name was too long to keep using. So this was the title of his paper where he told the world that there's something that this tobacco disease is caused by a contagious living fluid, an infectious living, something liquid. He didn't use that term in the paper because it was too long. He called it something much smaller. He used a much smaller word. He used the Latin word for poison, which is virus. And this was the world's first time that people realized that diseases are also caused by something that are much, much smaller than bacteria themselves. This was the world's first report of a virus. Although, of course, people wouldn't accept this. Bajring was a lot like Mendel, Gregor Johann Mendel. They didn't realize the value of his work until much later. He was alive by the real credit to his work came only after 1950s when people realized the true extent of what he has done. But he was the first person to report that there is something smaller than a bacteria that can cause disease, right? So I showed you this. I said that that's my finger, that's magnified 4 times, that's my index finger magnified 50 times, and we still can't see bacteria. Now, and so I want you to picture this again to show you how small viruses are. I showed you this picture of some famous bacteria. I'm going to take this image of Mycobacterium and enlarge it. Okay, that is the enlarged image of Mycobacterium tuberculosis. If this is the size of a bacteria, that's how small a virus is. That's most viruses are about that small. You can compare it with the entire thing that I showed earlier. So you can't see a bacteria there, and that's how small compared to a bacteria a virus actually is. Now, which begs the question again, how does a virus live? What does it do? Bacteria and viruses differ very systematically in one important aspect, which is a bacteria needs food and divide. A virus's sole purpose in its life is just to make more copies of itself. It just wants to propagate. It's the smallest known, I don't know if I can call it form of life. It's at the very border of life and death. And it's very simple because let's say a virus infects, this is a cell, a host cell. All that the virus will do is once it infects the host cell, it has its DNA or RNA, and that contains instructions on how to make new viruses. And the virus essentially hijacks the cell. What it does is it forces the cell to make more copies of itself. It will tell the cell, you stop doing your work, you do my work, make more copies of me. And once the virus hijacks, the cell has no choice. It will keep making more and more copies of itself and releasing them into either the bloodstream in case of HIV, or if it is SARS-CoV, you can sneeze and it will release it into the atmosphere, as aerosol droplets into the air. The interesting thing is the only live component of a virus's life cycle is inside the host cell. Outside the host cell, it does nothing. A bacteria can be alive both inside and outside the host, but a virus can only live inside the host. Outside the host, it's as good as dead. It does nothing until it goes back and infects the host once again. So that's how viruses live their life. A virus uses the host cell's resources to make more copies of itself. That's all it does. It has one purpose, one objective in life, a virus, and that is just to propagate itself, to make more copies of itself. Viruses are generally host specific in the sense a human virus will not infect plants. It can infect other related animals, sometimes monkeys, sometimes it depends on the virus. For example, SARS-CoV-2 has been shown to infect tigers, even though cats actually, they don't cause disease, but they can infect. Humans and cats are not that far, we are both mammals, but it cannot infect a plant. So similarly, HIV, the human immunodeficiency virus causes disease only in humans. There's a related virus called SIV, simian immunodeficiency virus, that causes disease in monkeys. But SIV does not infect human and HIV does not infect monkeys. Viruses are very host specific that way. Similarly, vice versa is also true. A viral disease of a plant cannot affect humans. A plant disease virus is a plant virus. It cannot jump. A bacteria is not like that. A bacteria that causes disease in a plant can possibly cause disease in humans also. It won't. Most of the cases it doesn't, but it can. Lastly, sometimes related animals, like there are, like we are cousins with chimps, like that there are related viruses also. There are a lot of viruses in the pox family. There is cow pox, there is horse pox, chicken pox, smallpox. All of these are all related. If you have immunity to one, you can get immunity to the others. Of course, cow and a horse won't infect humans that much. They're very mild disease they cause. So coming to a direct comparison of a bacteria versus a virus. A bacteria, like I said, can stay alive both inside and outside the host cell, whereas a virus depends on its host for survival. Outside the host, it's as good as dead until it infects again. Antibiotics are very effective against bacteria. They don't, I mean, you can get an infection because they live outside also, but you can treat them. Today, bacteria are very compatible organisms, but viruses are not. Those antibiotics are specific to bacteria. There are a small class of drugs called antiviral drugs, but the problem is an antibiotic, if I take, it's going to work against a big group of bacteria, whereas antivirals are very specific. If I take an antiviral for HIV, it's not going to protect me against SARS-CoV-2. Now, a lot of reasons for this, why we cannot make one broad antiviral agent. The biggest reason is that the virus uses your host machinery to make copies of itself. So whatever you make has to be specific to the virus, not to host. If you remember Paul Ehrlich's magic bullet, we need something that can attack only the virus and not the host. All existing antiviral, the maximum amount of research has been done on antivirals only for HIV. Now there are there's research being done on SARS-CoV-2 also. But even with HIV, we don't have what is like the Ehrlich's concept of a magic bullet. What we have is something that will prevent the virus from multiplying, but it will also cause a lot of side effects. So we don't have a lot of medicines against virus. The only way is wait for an immune, treat the symptoms, wait for the immune system to make things better. But this point about the virus, where it depends on the host for survival, is actually very, very useful for us. Useful how? Because it means that viral diseases can be truly eradicated. What do I mean? A virus needs the host to make copies of itself. If every person on the planet is vaccinated against a virus, then the virus has no host. Okay, it's not going to go and infect another animal. It can't do it. It's host specific. And if we manage to vaccinate every single individual, then the virus has no host and the virus is gone. That's what happened with smallpox. I don't know if many of you know this, but anybody who is born after 1985 has not been vaccinated against smallpox. I haven't got the vaccine. Many of the students watching will not. The older people born before 85 would have got the smallpox vaccine, because at that time smallpox still existed. But by 80s, WHO said we don't need to vaccinate because smallpox is gone. I'm sure Dr. Jayaprakash mentioned this in his talk. The last smallpox vaccine was given in India in the 80s. There's no need to vaccinate again. There are a few vials of smallpox stored in certain highly secure facilities in the world. This is just in case by some hook or crook it reemerges, we can immediately make the vaccines again. That's why that is kept, right? So now there's a very interesting question that many people ask me with viruses, which is, if a virus really needs its host for survival, if a virus cannot live outside of its host, then why does it kill the host? If it kills the host, it's not going to make more copies of itself. So why would you kill? It seems to be, it's like you're cutting your own hand that feeds you. It doesn't make sense. The reason is actually the death of the host. Even the virus doesn't want it. The virus doesn't want to kill the host. It doesn't want to kill you, but it happens because your cells are so tired. Your cells are exhausted by making more and more copies of new viruses that they can't help but die. The death of the host is an unfortunate consequence. Even the virus does not desire it. But then that's how evolution works. How does the virus survive? It's very simple. The virus ensures that before the host dies, it has to move on to a new host. That means that if a virus has infected you, there are two conditions here, not just dies. It can also mean the immune response takes over and immune system goes and controls the virus. The virus has to spread before either the immune system controls it or the host dies. You can see this proof of the statement is the fact that the viruses that spread very easily, like SARS-CoV-2, influenza, Ebola, Marburg, all of these viruses will kill also quickly. That's because they have, when I say kill quickly, that means in a span of 2 weeks, 3 weeks, something like that. Compared to HIV, which takes years to kill you, HPV, HIV, these viruses take years to kill you, and they don't spread that easily. They are very sophisticated ways of transmission. If HIV was airborne, humanity would have been eradicated by now. These viruses that kill quickly also have to spread easily because they've evolved in such a way that they have to spread before the host dies. And that's why viruses that have such a high infectivity are the ones that easily make pandemics. You would have heard of Ebola. Fortunately, it's not become a pandemic because it's very quick to kill and you can know symptoms very easily. But influenza and coronaviruses, they kill quickly and they also spread very easily. Simply aerosol droplets and airborne transmission is a very good example for this. Yeah, like I said, viruses that take their time to kill have very specific modes of transmission that don't spread easily. Now, if this begs another question about viruses, which is that how do they come from? From where? I mean, if a virus needs a host for survival, how did the first ever viral infection happen? Well, we don't know how the first one happened, but we know that most viruses are transmitted to humans through zoonosis, which is transfer from an animal. I know that most of the viruses which we do encounter are not going to be causing big disease, but some of them do, like SARS-CoV-2. SARS-CoV-2 came to humanity only because of the ways we ended up doing deforestation and all of those things. Last few years, we've been hearing of some new name of virus or the other. We heard of Ebola came up in our vocabulary, Zika, Nipah, SARS-CoV-2. All of these diseases are coming in only because of this one process of zoonosis, which is basically, the process of us getting a virus from a related animal, and it's happening only because our increasing cutting of forests and frequent contact with the natural world. Which brings me to my final question: how and why do bacteria and viruses cause pandemics? Well, the answer is actually very simple, and the answer to both questions is the same. Life always creates life. It's the most important rule for anything to be classified as living, that it should propagate itself. We human beings sometimes come in the way of the propagation of some other organism's life. Like, for example, SARS-CoV-2, if it has to propagate, it has to go through us. We come and we become an intermediate of that part of process of life creating life. It's an unavoidable part of nature. We cannot question it, and they're infecting us is the way some bacteria and viruses just propagate. It's like I said, it's the only rule of life. It's in their DNA to infect, and they've been on Earth for far longer than we have, and they will remain for much longer after we have gone. They infect us because they have to, but only if we give them a chance. They will infect us only if we continue to loot and plunder Mother Nature the way we have been doing all of these years, cutting trees and all. Blaming bacteria or viruses for the pandemic is like blaming a dog for biting you just because you threw a stone at it. So when people ask me how and why do bacteria and viruses cause pandemics, I say only one thing: they don't. Humans do. Thank you very much for being such a nice audience. I will be happy to take any questions.
All right. Thank you, Mr. Panchapakesan. We do have a few questions for you. The first question is, where do viruses originate from? And the question was asked by Aditya. So where do viruses originate from?
It's a very interesting question. Viruses have been, we really don't know how the first ever virus came from, but we do know that the primitive forms of life, when life evolved on Earth on something like viruses. So that's a very difficult question to answer. I know how human viruses come from. It's like I said, it comes through zoonosis. Almost all viruses that infect us today have originated in one of some animal or the other. It will either be in case of Ebola, SARS-CoV-2, all of these, bats are the source of these viruses, but they can be other sources. Bats, of course, there's an interesting connection between bats and these viruses. That's why Ebola, Nipah, SARS-CoV-2, all of these viruses come from bats. So most of the time, yes, it's zoonosis. It comes to humans from animals.
Question is from Avani Bhatt, and she's asking, what is that?
Bacteriophages.
Can bacteriophages help in fighting bacterial diseases?
That's a very interesting question. Yes, they can. In fact, there have been one or two reports of what is called bacteriophage therapy. For those of you who don't know, bacteriophage is a virus that infects bacteria. So if there's a virus that infects only bacteria, I said viruses are both specific, so they cannot infect humans. There have been cases when antibiotics have failed, one or two cases where they had no choice, the patient was dying and the doctors approved the use of bacteriophage therapy, but it's not yet approved for large scale use. There have been there's one case that I know of where bacteriophage therapy did work. The bacteriophage was introduced into the patient and it went and killed the bacteria that is responsible for the disease. I think it was meningitis that they used for the cure. I'm not sure.
Thanks. The next question is from Isha, and she's asking if reptiles contain salmonella.
They can contain. It's like I said, bacteria are literally everywhere. It's very difficult to say where they come from. Yes, but I know that reptiles do contain salmonella. Some of them do, yeah.
All right. The next question is, can viruses survive in different climatic conditions?
The time of survival varies definitely. If it's too hot, I mean, when I say survive, inside our body, the temperature is always going to be 37. That's maintained. But let's say I sneeze or something and like a few droplets fall on the surface, then yeah, if it is too hot, that the viruses there would die faster than on a surface where it's a little colder. But in either case, in both the cases, we know that those viruses are not the real danger. The real danger is the direct inhalation of aerosols or respiratory droplets that I'm actually sneezing out. So the climatic condition matters only if the virus exits the body. Inside the body, it's always going to be 37 degrees. That's the best for all the viruses that infect us. So, yeah.
All right. Sulan has a very interesting question for you. He's asking if bacteria and viruses are harmful to us, then why are they used to cure diseases?
They are, I mean, it's like I said, it's not all bacteria are harmful and not all viruses are harmful. But we have been consuming curd, lactobacillus, for I think thousands of years now. And there's no report of lactobacillus causing any infection. They're commensals, they're useful bacteria to us. Some bacteria are useful, not all of them. Viruses, of course, we don't really use viruses to cure. The exception, of course, is the bacteriophage therapy I said. We generally use viruses more in academic study. They're very useful to us as scientists to, infect and study. But viruses we don't use as such for, other than for vaccines, of course, we don't use for fighting any disease. Yeah, so not all of them are bad.
Right. Thank you. Ritik wants to know, what new things did you find in your research on the HIV-1C or IC?
1C. Okay. What we found was that Professor Jayaprakash spoke about mutations. HIV-C, we found out that the family of viruses that infects India, it creates more new kinds of HIV viruses much faster than the other families. This is a problem because as you have more and more new variants of HIV emerging, it's going to cause a lot of drug resistance and other problems that continuously are going to face us in the in the drugs that we have. We need to keep developing new drugs because the virus will keep getting resistant. That's one of the things that we found, that the Indian virus can make new viruses much faster than the other families of the virus can. So that's what we found out.
Thank you for sharing that with us. The next question for you is, if COVID-19 vaccination is given to everyone, will the virus die? The virus will die, but what will happen if COVID-19 can survive within animals? And will scientists give animals also vaccination? And this is from Arjun.
Yeah, okay, so I want to say something about the COVID-19 vaccines. The vaccines that we have got right now are all, they've been developed in record time. No other vaccine has ever been developed in the time that these vaccines have been developed. And the objective of these vaccines is not to prevent infection. All of these vaccines will reduce severity of the disease. It doesn't mean that you won't get COVID-19. It only means that if you get infected, your body can fight it much better. So of course, it's not that vaccine research on COVID-19 has stopped. They're still continuing. Every single industry and lab in the world, even our own lab, even though we specialize in HIV, we are also working on COVID-19. To answer your question, yes, if everybody is vaccinated, then yes, COVID-19 pandemic will end. If everybody has antibodies against COVID-19 in your blood, it can be stopped. Of course, there's always having said that, there's always the possibility that the virus will mutate and become endemic. When I say endemic, it means it will be localized to a geographical area. This town or this village will have it, and from there it will be in small pockets. That possibility exists. Vaccinating animals, it doesn't infect dogs, which are the most at-risk animals are our pets. It doesn't infect dogs. And cats, yes, it has been known to cause a few infections in cats, but again, I don't think it can spread from cat to human back. So I don't think they'll be vaccinating pets against the disease.
Thank you. Of all the questions that we got in our chat box, there was one comment for you as well, and that's that you look like Dhoni. And we move on to asking you the last question for your session, which is, like good bacteria, do we also have good viruses? And just another question. So the first question is, like good bacteria, do we have good viruses? And also, what do you advocate, prevention or vaccination?
Vaccination is a form of prevention. So, I would, if it's always better to go and get vaccinated, for the simple reason that you may be able to, you may be healthy enough to fight a disease, but the person who may be infected from you may not be as fortunate. That's what I would say for the prevention aspect also, which is that we really need to remember that it's not just us. All of us, the young people, are not the ones who are the at-risk group for COVID-19. If we take it carelessly, the mortality rate, the death rate for young people is very low, but it's always better to prevent it, because you may get and then you may spread it to somebody who may not be as lucky as you are. So that's why prevention is very important. Wear your masks, all of that, follow the precautions. And of course, I know that majority of this audience is very young. By the time we get our turn for the vaccination, probably COVID-19 will go away. But if it doesn't and you have a chance, please go get yourself vaccinated.
Yeah, just that last question for you was, So I'm just going to read the question out. So as you said before that most viruses spread through zoonosis, then research has shown that most ancient viruses are preserved in glaciers, and due to the rise in global warming, the viruses can spread after these glaciers start to melt. So can we say that we are inviting new pandemics as a result of our activities? And this is by Amogha from Nandini Layout Parikrma.
Yeah, your point about climate change being linked to the emergence of new pandemics is very true. Although it's the glacier aspect is unlikely unless a scientist goes, and it's like I said, the virus needs a host, and without the host, it's dead. Unless we have some, some mummy or something preserved in the ice, not mummy, some ancient Neanderthal or something preserved in the ice, and we go and dig up his body and do a post-mortem, and then from there we get it. That's a possibility. But the point about climate change, global warming causing pandemics is very, very important. There's already research suggesting that SARS-CoV-2 actually came in because of global warming. They said that the bats actually which caused this, the origin of the SARS-CoV-2 pandemic was because of the change in the habitat of these bats. They don't naturally live there. They flew there because of climate change. So climate change is a very important problem, and yes, it can cause more and more pandemics. I think I missed one question. Somebody said, "Is there a good virus?" There is. If for us, for scientists, yes, there are a lot of very useful viruses which we use in our work. I mean, let's say we want to put a gene or something. Viruses are very good at that kind of thing. So we use it. They're good viruses for us, but for the common public, probably no. I mean, the phage therapy, if it comes in, yeah, then they could be good viruses, but as such, no.
All right. Thank you so much, Mr. Panchapakesan. Your presentation really, really helped us in understanding the biology of bacteria and viruses, and for the overview of how bacteria and viruses work, and for helping us understand that we call ourselves human beings, but we really are a big blob of bacteria and viruses walking around. Thank you so much for
Thank you.
that viewpoint.
My
Thank you
pleasure.
so much for giving us the time of day.
Thank you so much.